We are very lucky today to be joined by Dr. Waseem Qasim. The title of the presentation is How to Train Your T Cells for a Battle Royale. Dr. Qasim is a pediatric immunologist and a professor of cell and gene therapy at Great Ormond Street Hospital. He trained in Newcastle and qualified in medicine with a specialization in pediatrics and immunology. And after conducting PhD research in T cell engineering, he delivered gene therapies from bench to bedside. His first in human applications of viral vectors were followed by trials in genome editing with TALENs, CRISPR, Cas9, and base editors. And now clinical trials are actively exploiting engineered CAR T cells against acute leukemia and overcoming allogenic barriers to provide universal cell therapies. So we are very excited to have you here today, Dr. Qasim, and I look forward to hearing your lecture. And with that, I'm going to turn it over to you.
Okay, well, good. I guess it's good morning for most of you. It's good evening here. Delighted to accept the invitation and to fill you guys in on where we are with some of our T-cell immunotherapies. Most of this is in the context of hematological malignancies, and I'm aware the audience is a mixture of postdoc students and clinical researchers, so hopefully the fit will be about right. I'm going to switch off the video, I'll switch it back on for questions, and then I'm going to run the slides, hopefully without any problems, but you obviously shout if you can't see anything. So, well, I'm based at Great Ormond Street in London, which is an NHS hospital. It's affiliated with UCL, which is the university. And, okay, a couple of disclosures just to highlight. We've had previous research funding from some companies, and some of their data will be mentioned, and there's some patents filed around some of the activity that's ongoing. That said, the title was drawn from, well, it's stolen, it's plagiarized from a couple of books and games. How to Train Your Dragon is the story of Hiccup Horrendous Haddock III, a Viking who comes across a dragon's egg and then recruits that dragon for his battles. And I understand that's now movies and cartoons and so on as well. And of course, Battle Royale is a gaming term, and you'll see how some of that might be relevant, but it's a mix of the two, and apologies for anybody who doesn't play the games or read the cartoon books. But let's get going. So I'm going to talk about, I'm going to give you a quick overview and a summary of what's been going on at GOSH, Children's Hospital, over the past 15 or 20 years in terms of gene and cell therapies, and that will explain how we've got involved in T-cell immunotherapy, and in particular CAR T-cells. And I'll focus the talk mostly on what we're doing for allogeneic CAR T-cells and how some of the early genome editing technology has turned out to be particularly useful for engineering T-cells for use in children with leukemia. And I'll describe how we've gone from TALENs to CRISPR, and then most recently to base editing, and what that's allowing us to do, which seemed quite implausible even a few years ago, is now in a therapeutic phase for first-in-man testing. So that's the overview, and that's where we hope to end up in about, 40 minutes or so. I'll allow some time for questions at the end. So our centre we're in central London. We're part of both a hospital and a university, and so there's good cross-fertilization between labs and clinic, and also the things you need in between to pick up cells from the bench, develop them, design them and develop them, take them through translational steps, which involve almost pharmaceutical grade manufacturing in clean rooms, dealing with, in the UK, it's the MHRA, which is the equivalent of the FDA. And in the UK, we have GTAC, which is the equivalent of what used to be the RAC in the United States for approvals to do gene therapies. And then also there's a whole lot of logistics and infrastructure around how you might collect cells and get them back either to the same patient or to different patients. And that's why, with a background in immunology and transplantation, these are the types of patients that are being treated first with the new wave of therapies. So that's the location and the overarching ambition of what we want to do. Our primary set of conditions that we manage, now we go back, that we manage are related to bone marrow disorders, in particular, two groups of disorders. One around immune deficiencies, where there's a defect in particular pathway, in particular leading to SCID disorders, T cell, B cell disorders. And then there's the blood disorders, of course, sickle cell, thalassemia, other blood disorders like Wiskott-Aldrich. All of these have turned out to be amenable for collecting and engineering stem cells to give back to patients. And there's also some conditions where you might want to replace enzymes for some of the metabolic conditions. And these have been early targets for some of the gene addition strategies where vectors are used to add genes into cells. And of course, there's the whole issue of how you might pull out immune cells, T cells in particular, also perhaps NK cells, other cells, to engineer them to express either a chimeric receptor or a T cell, recombinant T cell receptor, to use them for adoptive immunotherapy against leukemia or against viruses. And the two approaches go hand in hand, but maybe some of the objectives are slightly different, in that with the stem cell treatments, we need a long-term, perhaps permanent recovery, a long-term repopulation of a complete compartment. With some of the immunotherapies, you might only need the effect for a shorter period of time for anti-leukemic, anti-cancer effects. But in both cases, we're involved in harvesting cells, manipulating them in a clean room. Cells are dividing, so any vectors that we use have to be integrating, or any change in the DNA has to be permanent. And then there's the important issue of how when we give the cells back, they need to have an advantage. So either that involves some chemotherapy or some other way of giving the cells an advantage. Sometimes they'll get an advantage if you're repairing a particular pathway and there's a survival advantage. Other times you have to try and deliver that advantage. And for the main part, the gene addition strategies have used integrating vectors, originally gamma retroviral vectors, and then over the more recent period, lentiviral HIV-derived inactivated viral vectors, which are in quite widespread application now. So here's a summary of what we've been up to in the last sort of 15 years or so. So the stem cell therapies started with treatments for SCID and for ADA-SCID, moved on to the conditions like Wiskott and are still ongoing. The T cell therapies, the very first T cell therapy we made at Gosh was a suicide gene into donor T cells to prevent GVHD. We've done some TCR transfer work for viruses and for WT1, but the bulk of it has been around CAR19, and there's also studies running across London and elsewhere for CAR19. There's some GD2 stuff that's happened. And then in the more recent times, we've had UCAR19, which is a TALEN-engineered product. We've got CRISPR-Cas19 for B-cell leukemia, and we've got some base-edited products which are coming through now. And then also, just to say, we've got the same vector platforms that have been applied for other conditions. Here's an example at the bottom here, skin conditions for EB and for Netherton's, as proof of concept that the same vectors can be used in other tissue types where there's ex vivo engineering of cells and return to patients. So that's what we've been up to. And the advantages of these kind of strategies, which are all involving ex vivo engineering, is to be able to pull out the exact populations you want to modify. You don't have to worry so much about in vivo immune responses or biodistribution problems. And you can, if you need to, use the period where the cells are ex vivo to give chemotherapy and remove competition. So all that's helpful. The disadvantages, of course, are obvious, that you need a lot of infrastructure around this equipment to people that can do the manufacturing, clean rooms, the logistics of moving cells and taking them backwards and forwards. And of course, you may not actually be able to collect and target the exact populations you want, because you may not get the progenitors, the long-lived progenitors in your collections, or they may not survive the ex vivo manipulation, which is which is also possible. But having said that, these are the kinds of therapies that have gone into early phase studies that are showing quite a lot of promise. So the added bit of information that I kind of alluded to was we do need to just consider that the cells will go back into patients, whether it's a CAR T cell or a stem cell, and there's going to need to be some immunosuppression. There may need to be myeloablation if it's a stem cell niche that needs emptying, and there may need to be lymphodepletion, actual removing of lymphocytes rather than just suppressing them to allow these cells to have an advantage. And so we can kind of map how intensive these procedures in theory need to be and what kind of chemotherapy drugs are going to be involved. And for the purposes of today, we're talking about T cells, well, usually using fludarabine and cyclophosphamide, 2 chemotherapy drugs, with or without added serotherapy, which is antibodies against T cells and other immune cells. We're not really using any busulfan in any of these protocols that I'm going to talk about, but it is a relevant issue for some of the other products. So let's focus on T cell therapies, and you'll all be familiar with this now. For the last 10 years or so, some of the CAR T cell treatments have had some quite impressive results, especially in the treatment of leukemias and lymphomas in the B cell compartment. And you're probably familiar with this as well, that over time, all our leukemia patients have had better outcomes. When we go back 50 years almost, and the outcomes were 40% long-term survival. And over time, standard chemotherapy regimens have got better and better, to the extent that now we would expect perhaps 85–90% of patients to be managed with standard chemotherapy. But that leaves a reasonable percentage, 10% plus, maybe 15%, that will be in trouble at the end of their inductions or their standard regimens and will need extra therapies. And that usually involves going to bone marrow transplant. And in that context, we big centers will collect the most difficult patients. And on the right here, you'll see a graph from a summary of data that was published in 2017, where the outcomes of both B cell and T cell acute leukemia in patients that have had a transplant and then relapse is pretty terrible. It's almost futile to transplant these patients without some added intervention to try and get them into remission before a transplant. So it's these patients that we're focused on for some of the most difficult CAR treatments that we've got in play at the moment. So that's the context. This is just to summarize, and again, most people are familiar with this by now, that a T cell has an alpha beta T cell receptor, operates in a HLA restricted manner, whereas a CAR T cell has an artificial synthetic receptor with antibody derived binding domains. Those are going through the generations, even 4th generation ones are around. The talk that I'm going to show, the data I'm going to show is all around 2nd generation. They're all 4-1BB and Zeta, and they're recognizing antigen on the surface of target cells without any processing. And the response is not really physiological, it's supraphysiological, if anything. So CAR T cell studies, again, the widest application is obviously CAR19 and BCMA for B cell malignancies. And on this graph on the left, you'll see there's a whole host of other targets that are in therapies, and almost all of them are looking at the B cell compartment for B cell leukemia and lymphoma. And there's some applications coming through now for other types of blood cancers. And the numbers speak for themselves, that the successes have been in the liquid tumors and in the B cell compartments today. So this review from 2019 pulled out a number of limitations of the approach. There's obviously a risk of toxicity and cytokine storms. There's the risk that the cells may not perform as well as they should, or that the antigen is dropped and there's escape over time. So we can try and address some of these with the new editing technologies that are coming through. And of course, you'll also be familiar with Kymriah as the lentiviral CAR19 product that's most widely deployed. That's now a licensed approved medication, as are these other drugs on the table at the bottom here. There's now 6 of these in the US and in Europe and a few other places available as approved therapies paid for by the healthcare provider. So living medicines in patients available as almost like a standard of care. So how well do they work? Well, the immediate remission rates are very high. The longer term survival rates are improved, but there's still room for developing these and getting the numbers up to where we want them to be. These are, again, publications from a couple of years ago with larger scale readouts from autologous CAR-19s, where both products were showing improved survival and clear differences between patients treated with low disease and high disease burdens at the start. So the final thing to say on the autologous products, and the final lesson that's important before we go on to the edited products, is even with the switch from retro to lenti, there is still the issue of how these existing products with vector insertions behave in a longer period of time, and whether there is a risk of any transformational event or change that would be undesirable. And there's never been a transformation event with a lentiviral CAR product. There is clonal dominance, so certainly the cells are integrated, the vectors are integrated in places that are encouraging those cells to proliferate over a longer period of time, but no transformation event. And until last year, I would give this talk and I would say, well, there's been no transformation event at all in T cells from any manipulation, but that's not quite true, because last year there was an Australian study with a piggyback transposon system where there has been transformation of CAR-19 T cells, and these patients developed T cell malignancy with a as yet not completely understood underlying mechanism, but presumably related either to the piggyback transposase or some aspect of the manipulation that was carried out. So that just tells us that T cells, although are highly unlikely to transform, it's not completely impossible. And so that has to be in mind when we bring in the new steps that are going through with editing. So what do we want to do? We want to take allogeneic cells of a healthy donor, single donor, and we want to be able to use them in multiple recipients without the need for matching. So we want to produce an off-the-shelf CAR T cell that's already in the freezer, can be shipped around, don't need to match. You can time the infusion to the disease course and get an effect as impressive as with the autologous. That's the objective. So to do that, we need to engineer the cells, and we need to prepare the host, which I've talked about already in terms of chemotherapy and antibody therapy. So how can we deploy the various steps that are required? Need to remove the T cell receptor, because that's going to prevent allo recognition and GVHD. That's critical. So the cells must be devoid of T cell receptor. And we need to take steps to prevent them being rejected. Now, one approach would be to remove the MHC molecules, but that's not been the initial approach. The initial approach has been to remove this molecule, CD52, which is the target antigen of alemtuzumab, one of the conditioning antibodies. And so if we remove CD52, these cells are resistant to alemtuzumab and will have a survival advantage for perhaps 2 or 3 weeks while that antibody is in the system. And at the same time, we can think about other molecules like checkpoint pathways, PD-1 and so on. And we can think about whether the CAR is inserted with a vector or whether it's inserted using CRISPR homology repair. And we can decide what the editing is going to be, whether it's going to be TALEN or Cas9 or base editing. So the first approach that went to clinic was TALEN, and we got involved with a company called Selectis back in around about 2014, where TALENs against the TRAC locus, which is TCR alpha, and CD52, once provided as mRNA, turned out to be highly efficient at knocking out both those genes simultaneously. And the products that were coming out from that manufacturing were highly efficacious in the models, and so went early to clinic with the first patients treated in 2015. And this is the kind of manufacturing that was going on at the time. The cells would come in, they'd be handled in the clean room manually using these kind of devices. electroporated using something called a BTX electroporator, expanded in a wave bioreactor for a number of days, purged of any residual TCR alpha beta T cells using this Clinimax device, and then frozen in dose vials ready for use. Manual process taking close to 18 or 19 days and 3 or 4 GMP operators, so quite labor intensive. And the cells at the end of that procedure were characterized in detail, including this type of analysis, where we look for we look at the carrier type, and we look for any changes using FISH across the TCR locus. And occasionally you would see these kind of changes. So on the bottom carrier type plot, you can see there's a movement between chromosome 1, which is the site of CD52, chromosome 14, which is the track locus. So that's a stable translocation between the two editing sites. And those would occur at frequencies of, well, a few percent, less than 5%, but didn't appear to have any detrimental effect on the products. And so were documented and recorded, ready for any possible adverse event should it arise. And so the first application of those cells took this kind of form, where children with resistant leukemia who had been through transplant and relapsed were offered treatment on the basis that we would give some conditioning to reduce any residual immunity, infuse the cells as a single dose, allow them to work for perhaps 4 weeks, and then remove them if the patient's in remission, and proceed to an allogeneic transplant, ideally from the same donor as the original transplant. So this was a bridge to a second transplant, which would otherwise have been considered perhaps almost futile. So first 2 patients that were treated were reported in 2017 and had quite remarkable effects to just a single dose of the UCAR19 cells. And those 2 patients are still in remission some 7 or 8 years later. The product itself was then, or is, in larger phase studies picked up by another French company called Servier, and then Pfizer and Allogene. And their trials are mostly running in the US in adult lymphomas at the moment. But this study was published after we completed studies in children and in adults in Europe in particular, to prove the concept that these cells can be manufactured, preserved, and infused. But bearing in mind the patients that are enrolled on the studies are the ones that have failed all other options and are coming with quite a lot of free treatment and morbidities. So Talon's still ongoing, other investigation. Other platforms are also in play. There's a whole bunch of companies investigating other platforms, including meganucleases or homing endonucleases. Precision is running studies, again, mostly in the US, in B-cell malignancies. CRISPR Therapeutics is running a CRISPR version where they're inserting the CAR gene into the tract locus, and there's other studies running, including one that we've recently completed and published using a combination of lentiviral and CRISPR delivery, which I'll show on the next few slides. So once our Talon interest had been passed on, we as an academic unit picked up CRISPR, and CRISPR, of course, again, most people familiar, will allow very specific double-stranded DNA breaks, which will repair either by non-homologous end joining, and we can use that to knock out genes, or by homologous repair, and we can use that to insert genes in a particular locus in the genome. So our application, bearing in mind that the genesis of this project was perhaps 2016, 2017, at which point we didn't know how to manufacture compliant high-grade guide RNAs. So instead, we embedded them into the lentiviral long-term and or repeat. And that had a good advantage because it meant every cell that was edited was also expressing the CAR. And it meant, if you look at these flow plots on the right, it meant at the end of production, once we processed the cells, CAR expression was very high, above 90%, and editing frequency was also very high. And crucially, the residual TCR in these cells is down to 1%, which means the risk of GVHD is very low from the for a given dose of CAR T cells. So that product was manufactured again in our clean rooms at Gosh, and we moved from all those manual steps that I showed you on a previous slide to automated manufacture using this device on the left, which is called a Clinimax Prodigy. It's got a chamber that keeps the cells at temperature, feeds them, gives them CO2, and it's got a magnetic bead depletion system embedded in the system. And it does most of the manufacturing except for the electroporation step, which is done off device using this Lonza device, which we made compliant for the purposes of the manufacturing. So much easier manufacturing, down to perhaps 10 days and 2 operators as opposed to 18 days and 4 operators. And here's a summary of the kind of products we managed to get off for the clinical trial. You'll see high levels of CAR expression, very low levels of residual TCR, and then it has to pass a bunch of formal release assays before it can be used in the clinical study. The testing extends to looking for both on-target and off-target activity. And in the preceding work, we've done quite a bit of work looking for off-target sites using digenome analysis. And we picked out the top 6 sites that were likely to recur and screened for all of those by next generation sequencing. We set up something called a ddPCR, digital droplet PCR, to quantify possible predicted translocations. So those were mapped. We tested every batch. Some cells from every batch was tested in non-skid mice engrafted with B-cell leukemia, just to confirm potency was retained in a short clearance assay. So that's done for every batch. It's released. And then the study itself recruited children up to the age of 16 at the Gosh site with relapsed refractory B-cell leukemia for patients who were either unsuitable or had failed autologous CAR-19. And the treatment again was set up as a bridge to getting patients into remission and then onto transplantation if that was achieved. And in the chemotherapy combination, you'll see here fludarabine, cyclophosphamide, and alemtuzumab to give that advantage for the CD52 knockout. And so the first 6 patients or so reported just at the end of last year. And again, don't go through all the details on this slide, but it's usefully highlighted in a color scheme. So the gold is telling us that they'd had lots of previous therapy, including transplantation, previous CARs, previous bites. And all of them, the red is telling us across the middle, had residual disease that wasn't shifting ahead of a second transplant. And 4 out of the 6 patients that were treated there went into remission and onto allogeneic transplant using, where possible, the same donor as the first transplant. And again, the response is mapped in all of those patients. But there's a couple of useful things to pick out that you can see the cells. Here's an example. You can see the cells in the circulation within 2 weeks, picked up on flow here using Fab staining. And the cells are not expressing CD52 or the T cell receptor. And we can map the expansion and then removal of the cells using PCR techniques. And all the patients, you can see a peak around about day 14, which coincides with CRS and other things going on in terms of side effects. And so what we think is these cells expand in the first week, have quite potent anti-leukemic effects. And if they're going to get you into remission, that effect will be probably all done by day 28. So it's a short, deeper clearance compared to some of the other strategies that are employed perhaps in the autologous setting. So that's CRISPR and Cas9. I'm going to spend the next few slides describing how the platforms have gone from CRISPR to base editing. I'll explain what that is and where that is being applied for the first patients. So B-cell leukemia is quite widely catered for with a number of studies, very few studies running for T-cell malignancies, although some encouraging data coming from Singapore and from China showing that, well, perhaps this is something that is amenable to immunotherapy if we can get over the issue of T cell on T cell killing or refractory side effects. And so in T cell malignancies, there's a number of targets shown on this slide. There's some monoclonal antibodies that are in play, but we focused on CD7 as a useful target in the majority of pediatric T-ALL as a useful CAR target. And the technology we're going to use to allow us to edit cells to remove CD7 and stop fratricide, remove the T cell receptor, and simultaneously remove CD52 is called base editing. Came out of David Liu's lab at MIT 2016. At the top here, we've got cytidine editing, which converts by deamination cytosine residues to uridines, which when DNA is then replicating becomes a thymidine, so it's a C to T change. And we can use that to introduce stop codons or disrupt splice sites and prevent gene expression without the need for any breaking, any double-stranded breaks, which in turn has its own advantages. There's some follow-on technology where there's engineered synthetic deaminases to convert adenosine, and that is also of use, but isn't in this product, well, not at the moment. So I'm going to focus on cytosine editing and how we've managed to pick that up and take it through to phase 1. So this is what we want to do now. We want to take a healthy donor T cell. We want to remove the T cell receptor alpha beta complex. We want to remove CD7. We want to remove CD52. So triple editing without causing translocations, efficiently deploy CAR7 using a lentiviral vector and use those cells to target T cell leukemia. So what's the advantage of this? Well, one of the main advantages is if there's no breaks, the risk of translocations is very low. And so if we use ddPCR to compare multiplex editing, in this example, there's 2 edits going on, comparing base editor 3 against conventional Cas9. You see with the Cas9, there is low frequency translocations going on, perhaps 1% at the 4 possible sites, but those are all but gone with the BE. And that, as you put more edits in, becomes more important. So that's one key advantage. The second being the efficiency is actually very high. So just before the COVID close downs, 2019, we commissioned production of a lentiviral vector. We commissioned production of guides, which are now available synthetically at high purity. We generated an mRNA for the expression of the base editor, and we had in place agreements for supply of healthy donor mononuclear cells from the transplant registry. So with those ingredients assembled, we took them back to the Prodigy device and the same Lonza electroporator, tweaked the manufacturing process from what we were using for CRISPR with some changes, and manufactured the first base edited products using that system. And here's an example of one of the manufacturing batches. Again, CAR expression at quite high levels, knockout for TCR and CD7 and CD52 shown on flow at the end of production. And if we take those cells and we look at the DNA and we sequence across the sites, we can see in some of these cases 100% conversion rates at a very specific loci, introducing either those stop codons or disrupting a splice site at very high efficiency, confirmed on sequencing and to be at the sites as desired. And the graphs on the right tell us that all the editing activity is almost exactly where we want it to be. There's a little bit going on each side of that site, but it doesn't matter because we're introducing stops and disrupting genes rather than trying to have a highly, the fidelity of the repair doesn't have to be completely 100% as long as it stops expression of the gene. So that's the molecular signatures. We know the guides may take the editor elsewhere, so we need to look and think about off-target effects. We also know that some of these editors may operate and affect RNA as well as DNA, so there's a risk of promiscuous deamination both at the DNA and RNA level. So how can we screen this out and how much does it matter? We're going to use these T cells for a time-limited effect to remove leukemia and then destroy them. So some of this is maybe interpretable, and some of it is going to be difficult to know what it means. What we did do is we looked across the top 20 sites of predicted off-target activity and find nothing much above background in T cells at the end of production. And we looked across the entire binder sequence. We figured if there was ectopic stuff going on and it affected some of our binder sequence, that would be a bad thing, might redirect the specificity of the cells. But all this blue tells us here is this very low level activity through the manufacturing process at either the RNA or the DNA level. So we're confident there's nothing going on there, but this is not exhaustive and it's not completely comprehensive. It just tells us, answers questions for the sites we've asked about. It doesn't tell us about other things that may be happening where we haven't been able to look. Nonetheless, the product, highly efficacious. Again, each batch tested in animals, cells manufactured to a high degree of purity. So the roadmap in the UK to getting this approved is shown on this slide. And again, the details are not important, except it's a bit of snakes and ladders where you have to go through various approvals, both in terms of getting the funding, the hospital approvals, the MHRA and the REC, and then eventually your study can open. And the dose is manufactured as sufficient for the entire phase 1 study for pediatric TLL. And this is what that study looks like in reality. These are patients that have relapsed or refractory TLL, CD7 positive, and they undergo lymphodepletion after bone marrow assessment, receive a single dose of the CAR cells. Dosing is aiming to be around about 1 million cells per kilogram, but there's some margins on that are allowed. And then the patients are kept in hospital, monitored carefully and assessed after 28 days with a bone marrow examination, and if in remission, can be eligible and proceed to standard of care allogeneic transplantation, which is then under kind of routine monitoring for a 12-month period. So that's the agreed formulation. It's open in the UK to children eligible for NHS care. So I'm going to show you on this slide here what happened with the first patient that was treated, because this was then picked up by some of the news outlets. That patient, 13-year-old girl who'd had a transplant the year before for TLL, but unfortunately had relapsed within perhaps 4 or 6 weeks of that transplant, and then become resistant to all the chemotherapy drugs, and was the first patient recruited and treated in May of last year, again with the lymphodepletion, single dose of CAR T cells infused. We can track the cells by PCR. We see them expanding over the first month, and we see on the right-hand side the 3 colors showing chimerism, meaning signatures from the different parties. Originally, orange shows her chimerism. She'd already had a transplant some 6 months ago, so it goes green. After treatment, within 7 days, all circulating cells are derived from that CAR donor, and then that continues until she's in remission and is transplanted back to green from the original transplant donor. So that's quite nice to be able to track it using the sensitivity of the chimerism assay. And what happened to her disease, that's shown on this slide here. 2 different ways of measuring these diseases. So we use PCR, we use flow, and we like the 2 to be measured hand in hand wherever possible. Sometimes it's only possible to do the PCR if the quality of the sample is limited. But again, you can see going back 12 months, high levels of disease mapped on both techniques. Her first transplant got her into remission based on flow, but not on PCR. That disease then expands. And then 1 month before we treated, she's got high levels of disease, a single dose of cells infused, and is in remission with ongoing remission. This goes to 4 months, actually 6 months, and continued reconstitution at home out of hospital. So that study is recruiting. The next ones are going through now. We aim to treat 10 patients in the phase 1, try and reproduce this effect and see what the safety parameters are. The main issue has been the depth of lymphodepletion and immunosuppression, both by the drugs we give and the effects of the anti-T cell CAR T cells that are given in. So we're wary of viral reactivations and side effects from being very lymphopenic for a prolonged period of time. So as we stand at the moment, we have a CAR CRISPR study, which is treating B-ALL. We have a phase 1 for T-ALL, which is running now using base editing. And we have an application in for approval to start a AML study using a CAR33 base edited product, which is manufactured but not yet open. But that should then provide us cover for the acute leukemias, at which point we'll look to see which ones of these have got potential for further development. So that's the state of play. We started off talking about Battle Royale. So the aim is obviously to get to the end of that game, and at which point you get a crown and it's Victory Royale. But at this time, all we can say is that there is lots of technology around. These applications for first in human are helping us produce safety information and providing bridges to transplantation. And we've got early evidence of benefit from both the platforms that are being used in some wider applications in the pipeline. And I think that's my last, no, there's one more. So I've described base editing. I should also mention that there's prime editing in the pipeline. Again, David Liu's lab's come up with this, and this allows targeted gene insertion repair with synthetic DNA template engineered within a cell. And there's a whole variety of, ooh, what's happened? There's a whole variety of versions of how we can use this to insert genes into cells. So I think this will be the next thing to watch along the way, is how these can be deployed for further applications in immunotherapy. Last slide is acknowledging people in the lab, especially some of the funders, in particular NIHR, and collaborations along the way. And I'll stop there. And I will put this back on.
Thank you very much. Very interesting talk.
Yeah. Okay.
The questions are coming in, so I'm gonna jump right into questions if you're ready.
Yep.
The first question is, can you assess toxicities and possible side effects for the approaches using animal models before directly going into human trials?
Yeah, it's a very important question, and I don't know if I have a definitive answer, but I can share what we've done and what we've been MHRA has agreed we should do. So for these studies where we're doing T-cell work, the animal models are, there is animal work going on, and mostly we're using it to assess potency and confirm the immunotherapy effect. Those models are not particularly well designed. In fact, I don't really know of any good models to look for genotoxicity in T cells. So it's hard to have, without a positive control, to run those models, and that's not being required for these types of applications, for T cells. For stem cells, it might be different. There are models for stem cells where you can, if you trap them long enough, or you do serial transplantations after stem cell engineering, you could pick up transformation events. So that might be different. And it might be different in other contexts if there's a different gene going in, or the edit you're producing is more likely to cause some change. So I think the message would be it's context dependent, both in terms of the cells that you're engineering, the targets, and the potential of the model to pick up something that's going to be untoward. So that's where we are in the UK. It might be slightly different in the US.
Next question is, how long does the activity persist? And if it requires multiple infusions, does this have an effect on the T cell function? In other words, does it lead to T cell exhaustion?
Yeah. So again, a good question. So these particular studies are designed and dosed in a way that we try and elicit as much effect as possible in the first 2 weeks or 3 weeks. And the reason for that is we don't want prolonged lymphopenia beyond a period of about a month because of the risk of infections and cytopenias and so on. There have been some patients that have been redosed, not in our hands, but in other people's and adult studies. They've redosed patients where there wasn't expansion after the first infusion. They may have given a bigger dose or a dose from a different donor. So that is feasible, it's possible. There may be reasons why cells don't expand. There could be even though we screen out for HLA antibodies, there could still be an anti-donor recipient response. There could be a response against some component that we haven't been able to detect. Some of these engineering tools have got bacterial components in. Or there could be some other drug hanging around that we haven't thought about how long the half-life is. Some of our patients, for example, get inotuzumab or did, and that hangs around a long time. So all these things are reasons why cells might not persist. But the aim is to try and get their activity up and the effects within the first week or 2 weeks. That's how these are designed at the moment. They may change, and it may be that you want to persist for a longer period of time, and some of the editing may allow you to do that. At the moment, there's more questions than answers, I think.
If anyone else has questions, you can put it into the Q&A, and I have one here from myself. I saw that you were going to be expanding into AML. Have you thought about any other cancers like infant leukemia, or anything else on the horizon past AML?
So our focus in this hospital is pediatric malignancies. So acute leukemias, lymphomas to a lesser extent. There are collaborators who want us to help them with other types of blood malignancies, so multiple myeloma or other types of lymphoma, and those, of course, will be suitable targets. We're not. My group isn't doing any solid tumor work, not at the moment. There are other people that want to do neuroblastoma, for example, and there have been studies here for that. So anything where the targets are identified or well identified would be a reasonable place to start. Our own particular interest is the blood cancers, because that's where our background expertise is.
So how about the infant leukemia, like JMML?
Sure. I mean, I wouldn't. The numbers we see are very low, so there's no program. I wouldn't rule it. We're the kind of hospital that potentially could do one-offs or small numbers of compassionate use if there was a suitable target and it was already in play. That could be adopted. There is no program as such for JML, but it could be. I wouldn't rule it out.